we have to move on. We now continue with Alena. Back to the freshwater. See.
Yeah,
this one? Yeah, okay. Where is it?
Yeah, this is usually F5, huh?
Yeah, you go.
Or not?
Yeah, just wait.
Okay. Hi, there we are. One back. Hello, so I'm Alena Gsell. I'm a PhD student here at the NIOO with the Department of Aquatic Ecology. And today I would like to use this opportunity to take you a little bit into the realm of parasites and to entice you to think also about parasites when you look at the ecological functioning of your ecosystems. So if you look into time series data, there is not very much of it. But if you look for aquatic parasite, host parasite dynamics, then there are even fewer. There are a few for fish culturing, and there are also a few for phytoplankton and parasite series, but it is all very restricted. And to discuss with you why that could be, I first want to take you and introduce you to aquatic parasitism in general, and then I show you one case example of Astronella formosa, which is the diatom you see. This diatom you see below here, and it's a fungal parasite, the chytrid, which is this little bulb here or here. And then show you those examples and then see what we can actually learn from the time series. So why are these time series data rare for host parasite dynamics? Is it just because parasites are so rare? Because they have no significant biomass? Maybe they don't have an effect? Or is it just because they have such a hidden lifestyle and they're quite difficult to observe if you're not looking especially for them? And so that would mean, is there not enough sampling effort and maybe the methods aren't quite right? Or are these species just too cryptic? And maybe it's the best thing to first start with a basic definition of parasitism. So obviously a parasite inflicts damage to its host. It lives in an intimate association with its host or on its host, which is different to a predator, which is usually able to consume several prey items in its lifetime. So also the parasite has to spend a major part of its life on the host, and it derives almost all of its nutrition from only one host item, or maybe a few if you have parasites that live in longer cycles. And these ones would then also be often trophically transmitted. In general, but that's not necessarily true for the aquatic parasites, they are smaller than the host. They have a faster generation time, so they also have more adaptation and evolution potential. And in general, they impair host fitness. They don't necessarily have to kill the host, but they often do. So they lower competitive ability, because if you're sick, you can't function quite as well as you would usually. They might reduce survival. There are many castrating parasites, so they use up the energy that would go to reproduction of the host and use it for their own growth. And they can even change behavior. What you see, I mean, that's a terrestrial example, but rabies would be one example that you all know. So if you look for parasites in the literature, also in the aquatic literature, they are actually very common. Here you have a graph where you see different taxa, like here the arthropoda or plethelminths. And on the y-axis you see the proportion of species that have a parasitic lifestyle in these taxa. And as an aggregate of over all this data, about 40% of the species in each taxa have a parasitic lifestyle. So they're very common. Parasites are also abundant in numbers if you look for them. And this is an example from a study where Emily Lefebvre looked into small flagellates and ran a molecular study on them just to determine out of all those small flagellates in fresh water, who are they and what lifestyle do they have. And also here you see that a substantial part has either a saprotroph or a parasite lifestyle. So they are abundant in numbers. And if you then, she didn't look into viruses, but if you look for viruses like Burke and his co-authors did a while ago, you see that you have really amazing numbers. So there are numbers. And they also do constitute biomass in systems. So this is another example by Couris, who looked at biomass of free-living hosts and parasites in not fresh water, but in 3 estuaries. In this graph, you see biomass of different host species. Here you have the logarithmic scale of biomass density, and they're compared to the biomass of the winter bird population. In the graph below, you see how much of that host biomass is made up from parasite biomass living within those hosts. And if you then compare that again to the standing, use this one. If you use that then to the standing amount of birds biomass in winter, you see that parasites actually can scale up to the same mass as birds. And I mean, we know that birds have an influence on those systems. There is the same biomass of parasites, so why not? We also know that parasites can have as much influence on host population dynamics as predators or as grazers have. I use here an example that comes from the study system that I've been working on, again, the Diatom astianella and its chytrid fungus. These are 2 years where we followed spring bloom dynamics. So below here you have the first 24 weeks of the year. You have in black the host dynamics, which is cells per milliliter, and you have in white the parasite dynamics, which is a prevalence of infection, so the percentage of infected individuals within the total population. And this is just an example for 2 years where you see that you have a typical Astianella spring bloom coming up, and it is tracked by the parasite. And in our system, we know that silicate isn't limiting at the end of the spring bloom. So this demise of the spring bloom is really brought about by parasitism, which is, if you look at these numbers, can be very high, between 60 to 100% prevalence. Part of this dynamics can be explained by host density dependence by the parasites. So the more host there is available, the more parasite production you have. And part of these dynamics can also be explained by the environmental control of parasite activity. This is a study by Kees Bruning, who in the lab exposed Astianella and its parasite to different temperature and different light conditions. So here you have light and the temperature. And he was looking for the conditions in which the parasite cannot infect, so that's the black areas here. And for the conditions where the parasite needs either a lot of host or not so many hosts to actually start an epidemic. And what you can see here is that the parasite is both dependent on light and on the right temperature. So if you have absolutely no light or very low light, it can only survive in low temperature because then the whole dynamics is a bit slower. And the same also goes for low temperatures and high light. Can you actually see? Oh, okay, it is actually visible. So there is an environmental control of the parasite activity. Furthermore, parasites can also function as prey, which has been shown by Maiko Kagami and her co-authors, where you usually have the assumption that large inedible diatoms would actually be lost from the system through sinking and maybe then through biodegradation via bacteria and go over this way back into the food web. But via the fungal parasitism, you have those free-swimming zoospores that are very well edible for zooplankton. So in this way, nutrients from the otherwise inedible Acinella can be made available for the food web again. So that links into that parasites actually have a place in food webs, and they actually change food webs quite a bit if you do include them. So here you see maybe more traditional food web with producers and different consumers. But if you do add the parasites which link all the levels, you get quite astonishing changes in the topology of your food web. So that also means you obviously include more species, you increase the trophic links, you increase the connectance, and it also dilutes what you usually see, this kind of pyramid structure. It dilutes this pyramid structure. Yeah, I think I'll leave that because I'm a little bit low on time. Then I move on to the case studies. So these have been my pet subjects for the last few years. Again, the Acinella and its chytrid fungus. Acinella is a colonial freshwater diatom. It is ubiquitous. It is really abundant in many temperate lakes and rivers around the world. And it is a typical spring bloom species because it is a very good competitor for phosphorus. And it can also already grow at very low temperatures, like 1-2 degrees is already enough for it to start growing. Gsell planktonicum is the chytrid. It's an obligate parasite on Acinella. It can only live on one other diatom. And its infectivity is temperature dependent, but it's also, as I've shown before, light dependent. The parasite has an interesting life cycle, which has some importance for the results later. It can have either an asexual life cycle, as you see here. You start off with free-swimming zoospores that infect Acinella cell, as you see here, and then grow into an epibiontic sporangium, in which new zoospores are formed by mitosis, and then the cycle begins again. But in certain conditions, especially at the end of blooms and at the end of epidemics, but also in very cold temperature conditions, they actually go through a sexual cycle where you have a conjugation tube and a forming of zygote, which then forms a resting spore. And these resting spores are morphologically different from the normal sporangia, and they also stay inactive for about 3 months. So once this spore is formed, then the epidemic or the infectious development is actually stopped for the time being. Yes. Yeah. This is the location where we've been working since 1978. We've been, first Ellen started it, and then many other co-workers came in. We've been sampling Lake Maarsseveen regularly for host and parasite dynamic. These are the 11 springs that we could sample, so these are the same graphs as you've seen before. You have the first 24 weeks of the year, the parasite prevalence with the parasites in white, and the host density with the host in black. And you see that every year, pretty much, we've had a host bloom followed by a parasite epidemic. But lately, it has been we've been puzzled by blooms with no epidemic, or by a complete absence of bloom and epidemic. And we wanted to know, do we see a climate signal in these data? Yes. And indeed, we saw that there was a combination between how long the lag between the start of the bloom and the start of the epidemic is. So the start of the bloom, let's see a good example. The start of the bloom would already be starting here, but the epidemic only starts here, so you have a time lag in between. So the idea is that the longer this time lag between the onset of the bloom and the parasite epidemic, the higher the peak, the numerical peak of the host. And this is also then linked to the number of days that are colder than 3 degrees in the system. So the more days you have that are colder than 3 degrees, the higher is the cumulative host density. And combined with that, we do see a spring warming and a winter warming over the last, this is 50 years, and a decrease in contiguous cold days below 3 degree days in the system. We see that there is a loss of the cold disease-free growth refugia for the host. And the way that works, we think, is that in a warm year, you have host growth, the green line, but this growth is immediately attacked by the parasite, and they keep each other in headlock, which actually frees the competitive space for another competitor, which is the brown. Sorry, I actually don't see that. Which is the brown line. And in a cold year where you have the stop on the infectivity of the parasite because it's just too cold for it to grow, Arsenella can actually start growing and start building up a bloom and outcompeting its competitors. Whereas when the temperature rises, the parasite can become active. It finds a lot of host where the density dependence comes back. It can develop epidemically, terminate the bloom, and then you get the picture that we used to have of blooms and epidemics. So to just conclude from that very quickly, the general conclusions concerning parasites is they do exist. There are a lot of them, and I think that they have an ecological role, and it's worthwhile to keep them in mind if you find patterns that you can't explain otherwise. But what is maybe in this symposium more interesting is what we can learn from time series data. In our example, we do see that the host-parasite interactions are influenced by a changing world, so the increase in winter temperatures has an effect. We appreciate that there is a strong biology dependence of your system, so it really depends on the biology of your parasite, how that works out in your specific system. The parasite also is dependent on the environmental conditions, so there is actually a triangle of disease, the host, and the environment, and they all influence each other. Climate change may promote some diseases, but diminish others. So it is not, you can't only say that warmer is sicker, which is quite often a slogan almost. It is a bit more complicated in reality. And a lot of it is a result of changes in distributions and abundance of both the host and the parasite. And I really would like to stress that parasites are a functional component of ecosystems. And in some respect, they might even be a sign of a healthy ecosystem because they keep in check all the winners. And, of course, the thanks.
Thank you very much. Thank you very much for an interesting paper. Questions? Yes, please.
Yeah, I find it really funny to do it about this lake because I skate on this lake every winter for the last 3 years. And not in the 10 years before. Were you able to check your predictions that you know, with many cold days as we had in the last 3 years, you get a bloom and a
at least for 2010, that was the case, yes. 2011, I didn't sample anymore, but 2009 there was also a bloom and an epidemic. Then the really odd year was 2008, where we really had hardly any Asynella and any fungus in the system, but a lot of other diatoms.
And no winter as well.
No winter as well.
Any other questions? Yes, please.
Do you see any adaptations in your population?
Yes. What I do see is a genotype frequency shift in the Asynella population over 3 years, but that signal was more related to parasitism. So strong parasitism would then select for a more diverse Asynella population.
I found it interesting that you just told at the end that you think that parasites could be important to control species that could become very dominant. Is that actually the implication of what you said? So maybe are beneficial for biodiversity? Is that also an implication?
that is one of the theories that is discussed in literature. Yeah. There's also science for it, so that in a well-functioning ecosystem, you tend to have actually more parasites. And as you lose parasites, you can have an erosion of diversity.
Yeah, I'm more from the plant world, as you probably know. So there, we often have, if there is a strong dominance, that is negative for the diversity in the vegetation. So if the dominant, for some reason, is controlled, sometimes also by hemiparasites, for instance, that is often very beneficial for the diversity in the vegetation. So it could be a parallel here.
Yeah, absolutely. I think this is not only water, this is general.
Yes, probably.
Yeah.
Well, I also should mention that you are going to defend your PhD thesis next week.
Yes.
Right?
Yeah.
So that I will also be there, so that will be nice to meet
you have some questions still?
So, thank you very much again. Yeah. We will have now a lunch break, and I really ask you to be back here at 1:45, because the afternoon program should be scheduled even tighter than the morning program. Thank you very much.
Thanks.
Okay, well.